So Cindy Bell from Genome Canada will be moderating this next session. I'll be introducing her in just a moment. Just a couple procedural things for this afternoon. So, you know, we're going to try to be, the agenda has allowed 20 minutes for each speaker of the next 3 speakers to do their presentation and 10 minutes for Q&A. What we'll do is if the speaker sticks to that, we'll do the Q&A at the end. If they don't, what we'll do is we'll defer the questions to the end and we'll have the panel at the end. And again, the questions can then be addressed. So, so we'll just be timing things, and again, somebody will be making that indication here. So it's my pleasure to reintroduce Cindy Bell, who's going to talk about, just introduce the speakers for the next session, which is our applied session, and really focus on our CIHR partnership.
Thanks, Brad. So yeah, this is an interesting session in that not only is it applied, but as Brad mentioned, it's also projects that are done in partnership with CIHR, and of course the Forge project that Jen is going to talk about is a partnership with Genome Canada as well. And so the first person who's going to speak is Deborah, and Deborah's going to talk to you about the Vaginal Microbiome Project. And I think this was an initiative that, when did you launch this with CIHR, Brad? This is 2 years ago? 2 years ago now. So it would be great to hear the progress on this project.
Well, good afternoon. Thank you very much for inviting me. I'm really pleased to be here, and it's been a real pleasure to work with Genome BC over the last little while, because it's allowed me to see a number of other different projects in areas not entirely related to health, or even in the area of health that I work in. So I think for you guys, given what I saw for the morning, this will be something a little bit different. We are focusing on health, and in particular an area of women's health that I hope I will convince you is particularly important to take genomic tools to help answer. So I'm going to talk a bit about a metagenomic approach to the study of the vaginal microbiome. I'm struggling here at 5'1" with this podium, but anyway. So the microbiome, as many of you I'm sure are well aware, is really the sum total of all the microbes and their genetic elements, which includes fungi, viruses, bacteria, and other microorganisms. And what's very interesting, both scientifically and a little bit philosophically, is that in fact we're outnumbered in terms of bacterial cells versus human cells about 10 to 1. So then the question is really, who's us? Is it just our human cells, or is it us plus the bacteria that reside with us? And so that's for some debate. As you may well know, there has been an international human microbiome project that's been going on for a number of years, and Canada has been part of this, in particular with focused funding for niche areas in the body that are the microbiome is particularly important to health. And so there are oropharyngeal work groups, skin groups, a significant number of groups working in the GI tract, but the urogenital tract is also of importance to human health. So what I would like to do is talk a little bit about the vaginal microbiome, just to let you understand why it's valuable to understand this better. It's clear that the balance of organisms in the vagina are really critical to health. And a normal vaginal microbiome is actually the best defense against pathogens. Whereas the converse, where the perturbation of this balance of organisms is disrupted, this is perturbated, then it ends up with risk of post-surgical complications, adverse perinatal outcomes that I'll talk a little bit more about. Currently, if you open OBGYN textbooks and you have a look at how we understand the vaginal microbiome, it's based on very basic culture-based techniques where we believe that 90 to 95% of the organisms are lactobacilli, and the rest, 5%, are made up of a whole mix of different microorganisms, of which they're sort of vaguely referred to, often just by genus, not even by species. So that's kind of the level that a medical student would be expected to understand. And our very, very sophisticated gold standard in the clinical arena is the gram stain. So literally, that is the standard of diagnostics in laboratories across the world, where a swab is taken, it's smeared, and relative proportions of morphotypes are coded by the technician, and there's a grading system, and you basically get a score from 1 to 10, and that defines as normal, intermediate, or bacterial vaginosis. And what you can see here is the normal smear is relatively low abundance of microorganisms compared to an epithelial cell with large gram-positive rods that are the morphotype associated with lactobacilli, and then a little bit more in the middle slide, and this is multiple different morphotypes and higher abundance of organisms, and that's bad. So there we are, that's our clinical standard right now. However, despite the simplicity of that score, it has at least allowed us to understand some of the associated adverse health outcomes with having an abnormal microbiome. So we know that vaginal discharge, bacterial vaginosis, will happen in about 1/3 of women in their lifetime. It's the most common cause for going to office visits, so very, very high cost to the healthcare system. It's symptomatically very distressing for many women and is associated with complications post-surgically. The other thing that we're understanding more and more, it's also associated with increased risks of sexually transmitted infections because we don't have our appropriate defenses, so increased risk of acquisition and transmission of HIV, increased risk of acquisition of human papillomavirus, the precursor to cervical cancer, higher likelihood of acquiring chlamydia, and pelvic inflammatory disease. Probably half of it's related to this disruption in the microbiome. The other thing that's actually quite significant is the association of the vaginal microbiome to prematurity. So it looks like the idiopathic preterm births, of which actually 8% of births in Canada are preterm and about 13% in the United States, a significant proportion of those are actually related to infection or inflammation that triggers inappropriate prematurity of labor and delivery. Unfortunately, although this is only 8% of deliveries, it's associated with 75% of both morbidity and mortality, and significant adverse neurologic sequelae, with these little preterm infants actually having often point matter changes that result in cerebral palsy and long-term consequences. This is just a diagram, just illustrating that organisms that are normally associated with the lower genital tract actually ascend and cause a subclinical inflammation between the chorion and the amnion, and this results in this inappropriately timed contraction and or rupture of membranes. So I think, as I've alluded to, the consequences of having this infection or inflammation in this arena results in both preterm delivery, which can result in sepsis, pneumonia, respiratory distress, and in fact, death in some cases. So you would think, well, okay, we know that we've got an abnormal microbiome, we just do our little gram stain test, and then we test women, and then maybe we treat them, and then it's going to make things better. Unfortunately, large-scale clinical trials have been done utilizing this approach, and they've failed miserably. So we believe that it's the wrong test at the wrong time, that women at high risk are not being properly screened with a more sophisticated, nuanced test. We're not able to detect microorganisms in particular clusters that may be causing problems where other clusters are not, and we're treating all women as if they're the same. So genomic methodology has really offered a new way of approaching this problem, where we can hopefully get a better sense of actually the mix of microorganisms in a little bit more nuanced way. So this is where metagenomics has come in, which has allowed us not to do the typical thing, which is organism A present or absent using genomic technologies, but actually look at a population of microorganisms, look at what's there, their relative abundance, and understand a community of organisms. This can be done with whole metagenome shotgun sequencing, or can be done with targeted approaches, which is really much more common. And so one can use either a phylogenetic marker or a functional marker gene and use that as your gene to amplify. In the particular case of the group that I've been working with across Canada, we've been primarily, but not exclusively, using chaperone 60 as our target. And the reason for this is that CPN60 is ubiquitous in bacteria. It can be amplified readily from any bacterial genome, and it is a little bit more informative than 16S ribosomal RNA because it allows us a little bit better ability to go to species and subspecies level, which in a microbiome like the vaginal microbiome is very important, where the dominant organism is one genus, Lactobacilli. So if we can't do species differentiation readily, that's quite problematic for us. Not so much in people who are working in the GI tract. So this actually came originally in trying to develop a family tree for plants. Actually, it started in that arena, and that allowed us to then understand that in bacteria and in humans, there are highly conserved, highly replicative sequences of CPN60 in any particular species. It's about 550 base pairs. So our approach, just very, very briefly, is that samples are taken, total genomic DNA is extracted, PCR amplicons are created, and prior to high-throughput sequencing, there was clone libraries generated, sequence data, and then obviously, and I'll talk a little bit about our bioinformatics. And then finally, we get a collection of sequences that represent the community members, and then the challenge over the next while that ourselves and others are working with is how do we define what these clusters mean and how we actually describe them. So as you're all, this slide is probably a little silly for this crowd, but next generation sequencing has obviously been a huge advance forward for many of us. In our case, we've been using 454 because of the length of the CPN60 gene, and we've been obviously able to go from very small numbers of samples and slow throughput to be able to do a significant number of clinical samples to really start to answer these questions. The other piece that we've been working with our colleagues in Saskatoon primarily have been trying to optimize our assembly approach to our operational taxonomic units and trying to understand how best to both identify the appropriate units, to match them to our library, and then actually try and get some sort of a sense of a pseudo or abundance, relative level of abundance, which is quite important in our microbiome analysis. So we can then export through this assembly into various existing tools for further analysis and relationship to the metadata. So I want to just tell you a little bit about our study group, which we call VOGUE, which is the Vaginal Microbiome Group Initiative. And we have a whole series of both microbiome, virome, and fungal analyses planned, but we primarily started with the bacteriome as our first sort of endeavor. We have colleagues who are beginning to work on transcriptomics because we both understand and we believe that not just the mix of organisms that is there is going to be important, but actually what they're doing with each other and what products they're producing. And really, we're still working on improving our bioinformatic pipeline to better interpret our genomic data. The other thing that's part and parcel of our study group is actually a social scientist collaborator who's trying to understand what women perceive about health in that area and what practices are commonplace in the Canadian arena. And then we're also doing surveys related to our clinical sampling to understand what practices are associated with both normal and abnormal microbiomes. So I just want to share a little bit of data. This is a study, a little CIHR study we had before our collaborative study, where we wanted to begin by just trying to understand if we're going to start sampling women, normal, with or without health issues, is the menstrual cycle going to be an issue? Can we just sample them any day of the week? Or is it going to be that certain times are going to cause huge shifts in the microbiome and we really need to pay attention to that in sampling? So we began by doing a small pilot study to have a look at what shifts occur in perfectly healthy women, not on any hormonal therapies, who are just cycling quite naturally. And just to kind of cut to the chase very briefly, this is a summary slide. Obviously all the organisms here, it doesn't matter who's there, but just to give you a sense of, this is the menstrual phase on the far left, follicular phase, periovulatory, and luteal, which are the 4 phases in the menstrual cycle. And essentially our take home from this is that we're really not seeing significant shifts in healthy women in the non-menstrual phases. So it is a bit of a reassurance as we move forward with some of our clinical trials that in fact we could sample, we're still recording time, but it wasn't going to be probably a huge factor. The other thing that we noticed from this is that there are some natural clusterings already in this small sample of healthy women that we're seeing, where the vast majority of women were clustering on this heat map with 3 different species of lactobacilli, Jensenii, crispatus, and inners, and then a couple of women who were completely asymptomatic and well with Gardnerella, and then some other outliers with more unusual combinations that we're going to begin to try and understand whether these are normal or women who are asymptomatic but may be predisposed to health complications. So we, with this little study, saw 3 primary groups. We found that multiple samples for the same women clustered together, so really looked like they were fairly stable and no apparent relationship between menstrual phase. And we're constantly looking at other factors like ethnicity to see if we're seeing differences there. So then our VOGUE study, which is the one that's been supported by our combination CIHR Genome BC grant, is looking at a whole series of clinical cohorts using both our CPN60-based bacteriome approach, in some cases trying to understand the virome relationship. Our fungal analyses are pending, and we're doing some subgroups of these with some transcriptomics. But we're looking at healthy women, healthy defined by no clinical symptomatology or diagnosis, women who actually are HIV positive, women who present with recurrent problematic vaginitis and clearly have an abnormal microbiome, and then a couple of cohorts of women who are pregnant, where we're looking both at normal healthy and women who have very high risk for preterm birth. And then, as I mentioned before, we're layering this survey piece onto it to understand actually women's attitudes towards this and some of the practices that may or may not be associated with changes in the microbiome. So just to give you just a little bit of preliminary data from early days yet, we've managed to enroll 210 of our first 300 women in our what we call 1A, which is our healthy cohort, and we have results on the first 84 that have been through the sequencing pipeline. And we've been able to have a look at some general profiling, although haven't done the relationship to the metadata analysis piece yet. But just to give you a sense in these next few slides, this is how we're starting to understand where healthy women fall out. So we see a group of our women, the largest group, who are clearly dominated by Lactobacillus crispatus, which we know is a hydrogen peroxide producing, high acid producing, very protective lactobacillus. Inners is the next most common, then Jensenii is less common and is not as good a hydrogen peroxide producer, so may not be quite as good as a protective organism. Gasseri is also even more uncommon. And then we have some women who have another genus altogether that is their dominant organisms in different subgroups, but in classic vaginal microbiology world, these would be considered bad profiles, and yet here we are, we're finding them in perfectly healthy asymptomatic women, and then a couple of women with just very unusual mixes that we'll need to better understand. So just as a very preliminary, we're not trying to over-interpret, but we're seeing about 6 distinct clusters. We have to look at the stability of these, but we hypothesize that personalized characterization of this will predict susceptibility to disease and reproductive health. Just want to show you again some preliminary data for our HIV cohort that one of my master's students has been working on, Daljit. And just to quickly show you 50 women that are HIV positive with a variety of genital tract symptoms and multiple co-infections, as you might suspect in HIV disease. This is how they break down. So what we see here is a small group with bifido, not a very large group with crispatus compared to our healthy group, some with Gasseri and inners, and then a fairly significant number with Gardnerella dominant microbiology, and then a whole other mix altogether. And then what we've been able to do is relate this to our clinical scoring system, where a low score is good and a high score is abnormal or BB. And we can see that that seems to at least relate appropriately to our genomic results, where the high scores that we would consider abnormal are also associated with really mixed non-lactobacillus dominant microbiome. And then the other piece we thought would be interesting, again, just 50 women, but relating to their immune status. So CD4 counts is our way of determining the immune status of an individual with HIV, regardless of therapy or not. And a high CD4 count is associated with optimal health, the lower, the more immunocompromised. So what you see here is just a trend in these highly treated individuals to a lower CD4 count being associated with what we would normally think of as an abnormal microbiome. So just some preliminary data that suggests that this may be a way of better understanding the relationship between the immune suppression of HIV and the vaginal microbiome. These women were also symptomatic. So this is also just some preliminary data on another study that was funded by the March of Dimes, but we think it will help feed into some of our pregnancy data that we're waiting for, because some of the pregnancy enrollment is still pending. But we were able to enroll women who, here in Vancouver, had unexpected, spontaneous, premature preterm rupture of membranes, which is really a bit of a clinical disaster, to be honest, and almost always associated with preterm delivery, usually with morbidity. So we were able to enroll women between 24 weeks and 34 weeks, with 40 obviously being the abnormal gestation, who ended up with idiopathic rupture. And we were able to get vaginal swabs at the time of admission, and then weekly, and then we're following the infants. And we hypothesized that specific clusters would be associated both with the event and with the tendency to result in either a short time to immediately go to delivery and or with significant infectious complications. So this is just a tiny bit of preliminary data that shows that this group don't look anything like our healthy women at all, which I suppose isn't that surprising, but it's actually a little worse than I had anticipated. So the sort of purpley color is our lactobacillus dominant individuals, with these are individual samples here. So a very small minority have lactobacillus dominans, and the rest of them have a multitude of other organism clusters that really, I presume, it may well have been what was associated with the predisposition to rupture and or the predisposition to adverse neonatal outcomes that we do see associated with this event. So this is just very early days of our raw data, but I thought you might be interested in seeing that. So that's just a little tiny flavor of some of the work that we've been trying to do. Much of it's very much in progress. A lot of the samples are still being collected, and many of the assays are yet to be run. But we really think that we're hopeful, at least, that we're going to be able to define clusters that are associated with optimal health outcomes and clusters that are associated with these adverse, both non-pregnant and pregnancy-related adverse outcomes, and develop diagnostics, for example, maybe a Luminex platform. We're hopeful that we'll be able to get a predictive test for preterm birth that'll be a heck of a lot better than a gram stain. We're looking forward to starting to sort out the relationship with different, the bacteriome and the virome. We believe they're not just sort of living there unrelated to each other. And looking forward to our transcriptomics data, where we can start to understand what these particular clusters of organisms are doing. And then ultimately better understand through our survey work what lifestyle practices are associated with the healthy microbiome, because we believe that although there may be an intrinsic host microbiome that you have to start with, we believe that environmental and practices can change that. And maybe probiotic therapy, rather than blasting with antibiotics to alter the microbiome, would be a little bit more finessed treatment approach. So this is some of the members of our team at our group meeting in Toronto a few months ago. We have a group that is from across the country. Emily Wagner is based here and helps coordinate with me. We have a number of clinicians that are OBGYNs, infectious diseases. We have social scientists, and our microbiologists and plant and Genomic scientists are primarily based in Saskatoon and London, Ontario, and then our bioinformatics participants are both here and in Saskatoon. So it's a really diverse group with very, very diverse backgrounds, and it's been actually great fun to work together. We have a number of trainees working with us as well, and supported by a variety of organizations. So I'll stop there, and I'd be happy to take questions if there's time.
Time for a couple questions? Okay, great. Thank
We
you.
wasn't too sure about where am I at here? Okay.
Any questions? Elizabeth?
I guess I was interested in the probiotic. Is that, even without your information, is that a strategy that has had any success in the past?
You know, although intuitively the notion of replacing the appropriate bacteria makes sense, and a lot of yogurt companies are trying to make money on this. Unfortunately, the organisms in the yogurt products are actually often cow organisms that actually don't adhere to the human vaginal epithelium at all. And so far, none of the commercial products have actually shown in any studies to be a benefit. But we still believe that the concept is correct. We just don't believe the products on market right now are correct.
Thank you. And another question?
Are there some theories of how people, other than maybe diet, you alluded to, that why this microbiome ends up so different in some women?
There's a whole bunch of theories, and some of using traditional microbiologic techniques that have been shown. So clearly there are some women that don't do anything unusual in terms of their practices that from menarche have a disturbed microbiome that just is problematic from square one with symptoms and just you can't get them normalized. Don't know why. And no other particular clinical factors. Then there are other women who appear to start out with an appropriate microbiome, but through multiple sexual partners, douching practices. If you go into the pharmacy and look in the aisles in terms of products women can access to disturb their microbiome, there's thousands out there. And so that's the kind of thing that we believe is causing a lot of the problems.
Maybe one more quick question, Brad.
Deb, can you just comment on vaginal delivery and the impact that the vaginal microbiome has on normal birth versus, let's say, C-section?
So that's been I don't know if people heard that. So the question is vaginal birth versus cesarean section, and you're alluding to the infant's establishment of its own microbiome. And there's a number more, actually, colleagues looking at the gut microbiome of infants and children that are looking at that. We're hoping to as well. There is sort of teaser data that would suggest that there may be some differences in the natural starting microbiome of individuals who were delivered via vaginal birth or cesarean section. But it's not quite as kind of pure as people imagine. Like a cesarean section isn't like lifting an infant out without touching the microbiome of the mother. There's kind of a little admixture that happens, and often there's labor before. So it's not quite as clean and set out. But I think it's certainly an area for future study to understand the initial microbiome that an infant starts with.
Thank you, Deb. So there'll be time for more questions during the panel. Thank you very much.